think we're going to make a start. First of all, thank you everybody for being here this afternoon to discuss my PhD work in preparation for my Viva next week. For those who don't know me, my name is Oana Ancu and I'm in the final stages of finishing my PhD here at the University of Roehampton. I'm excited to be here with you today to present my work. It's okay. To present my work related to protein ingestion, exercise, and also one of the most challenging concerns of our time, type 2 diabetes. So my title for my PhD is Whole Body Insulin Sensitivity and Changes in Skeletal Muscle Insulin Signaling in Response to Protein Ingestion and Exercise. Sorry about the noise, there's been a bit of double booking with the room. Before I start talking about my research, I would like to just take a moment to acknowledge and thank the people that I've had the pleasure to work with over the last few years during my PhD. Firstly, I would like to thank my supervisory team, Drs. Richard Mackenzie, Dr. Astrid, and Dr. Nicholas Horan. Also my research group who's been with me throughout different experiments, Fulvia, Diana, Richie, and Alex, as well as our collaborators from University of Exeter, with whom we did our second study that I'm going to present later. Special thanks goes to Professor Francis Stevens and Dr. Tom Jameson, as I said, from University of Exeter. Also, we had some other external collaborators, Dr. Nicholas Bird, as well as Dr. Nicola Guess. And special thanks to our entire technical team, but special thanks goes to Tom, Alison, and Ryan, who's been together with us in our trials. And some other students that have been working with me, such as Monica, and of course my participants who have devoted their time and also tissue in order to take part of my research. So special thanks to them. Now I'm just gonna give you a bit of a background of my research topic. So we all know that type 2 diabetes is not just a medical challenge, but it has become a global concern, and it's affected millions of people worldwide. And it's a complex puzzle, and it has multifactorial concerns that leads to it. And we know obesity is a central piece in that development. My PhD focuses on one of these pieces of the puzzle. More specifically, it focuses on the impact of high protein on glucose control. Well, dietary protein is not new to us, or the importance of it is not new to us. We know that it's part of a healthy lifestyle and it's been featured everywhere. The issue becomes more complex when we think about dietary or increased dietary protein. And while we know it's popular for weight management, it presents to us with a paradox because we see conflicting results in terms of their impact on glucose control or insulin sensitivity. And this is a key factor in diabetes progression as well as its understanding. So due to this, we have set as our research objective to explore some of these mechanisms that are affected by dietary protein in terms of insulin resistance and glucose homeostasis.
I am so sorry to interrupt, Oana. Hello.
Of my background research area, I would like to just discuss a bit about some of the benefits that are associated with high protein diets, especially during energy restriction. So we're talking about weight loss here. So one of the most important advantages of high protein diets is that it has the ability to promote weight loss while it preserves lean mass, such as skeletal muscle. And this dual action of protein is very important when we're thinking about weight loss, because we want to have a healthy type of weight loss. We want to lose fat tissue rather than skeletal muscle. So that's why it is very important.
You're not sharing your screen, apparently.
Am I not?
Sorry. I don't know how you share your screen.
Here we go. I'm sorry about that. I forgot to share my screen. So we were just talking about how it has this dual ability to preserve lean tissue while it leads to a reduction in fat tissue. The second benefit of high protein diets is that it has the ability to improve satiety. So people would feel fuller when they eat protein-based diets or meals. And this can lead to a reduction in the intake or the reduction in the overall caloric intake, which again, it's a positive thing when we're talking about weight loss. Another important aspect of high protein diets is that it has the ability to maintain resting metabolic rate even during energy restriction. So that means that even when we're losing weight, our body is still utilizing a healthy amount of energy while at rest. Another aspect of high protein diets is that it's leading to an increase in diet-induced thermogenesis compared to the other macronutrients. So that is referring to the amount of energy that is required to digest, absorb, and utilize those nutrients. And lastly, it leads to an increase in insulin production after a meal. And while we are keeping in mind these benefits of high-protein diets, the main question still is there. What is the effect of high-protein diets on insulin sensitivity? And what sort of mechanistic pathway does it follow? And that's what we're here to explore. From all the dietary protein, a group of amino acids, more specifically, the branched-chain amino acids, have important roles in both protein synthesis as well as glucose metabolism. These are leucine, isoleucine, and valine. However, some studies have shown that increased branched-chain amino acid concentrations have been associated with insulin resistance. But let's remember that association does not mean causality. And also there are other things that we need to consider. We've seen differences between different groups of people in terms of these associations. And also we need to consider other factors, such as physical activity level. When we consider physical activity level, then maybe this branched-chain amino acid disease association is not the same. So just remember while we're talking about this, that these relationships are quite complex. Talking about complex pathways, now I'd like to discuss a little bit about the insulin signaling pathway. Well, I move this a little bit. I'd like to discuss about the insulin signaling pathway and how amino acids come into play in this signaling. So once the insulin signaling pathway starts when insulin binds to the insulin receptor. And that basically triggers a chain of reactions. Once insulin binds to the insulin receptor, then tyrosine is activated, and then insulin receptor substrate 1 and 2, so IRS1 and 2, come into play as well. What's happening is that they activate PIP3 via PI3K, and also they create that PIP3 from PIP2. And this is quite important because it sets the scene for AKT activation. AKT is also called protein kinase B. You may see it in different papers. And AKT is a very important component of glucose uptake in the skeletal muscle, but its activation is quite complex. It's quite complex. And it is complex because on one side it requires phosphorylation at the threonine 308 site, which is done via PDK1. But on the other side, it also requires this phosphorylation and activation on the serine 473 site as well. And this is done by the mTORC S6K1 pathway. And I'm going to discuss more about the mTORC pathway in my next slide. But now let's go back at the AKT phosphorylation. So once it is phosphorylation at both of these phosphorylation sites, it becomes fully activated. And only then it can phosphorylate its substrate AS160. And that basically works like a metabolic switch because what it allows GLUT4 to translocate to the cell membrane and allow glucose to enter the cell. we were talking about mTOR and we were talking about AKT. So we said that in order for AKT to be fully activated, it requires activation of both of those phosphorylation sites. One of them is activated by mTOR. But in order to activate mTOR, or what is a good activator of mTOR, is dietary protein-derived amino acids. More specifically, a very potent activator of mTOR is leucine, one of the branched-chain amino acids that I've mentioned. And mTOR has multiple functions. It's a central regulator of cellular processes. It's an important component of anabolic processes and cell growth. So when we're talking about protein synthesis. But again, the results when it comes to mTOR, they are different. So when we are looking at studies that are investigated isolated amino acids, infusions or ingestion, it normally leads to rapid and transient mTOR activation, which is slightly different from mixed meals and protein-rich foods. But we do have less studies that are focusing on mixed meals compared to the isolated amino acids. So with this observation and also the observation that we see an increase in plasma branched-chain amino acid in insulin-resistant people, we hypothesized that a chronic or excessive activation of the mTOR S6K1 pathway may contribute to insulin resistance in the skeletal muscle. And we have developed some interesting models to look at that due to the conflicting background that we have in terms of what's the impact of amino acid supplementations. If you are interested to find out more about the different topics, then feel free to read our review paper that was published a few years ago now. So this leads us to my first PhD study entitled The Effects of Acute, Moderate, and High Protein Meal Ingestion on Insulin Signaling in Skeletal Muscle and Insulin Sensitivity in Individuals with Obesity. Individuals or participants arrived in our labs in the morning. They arrived in a fasted state, and we have taken a baseline blood sample and a baseline muscle biopsy. After that, in a randomized double-blinded fashion, the participants had either 50 grams of protein or 100 grams of protein or 50 grams of protein with added fat in order to match the caloric content of the 100 grams of protein trial. So the last trial was a control trial because we wanted to see if the changes that we're going to see in the blood and in the muscle are due to increased protein content or due to increased caloric content. So remember that acts as a control trial. The protein meal was a chicken-based that was cooked in the food lab upstairs with the help of my colleagues. After they had the meal, we then started a labeled intravenous glucose tolerance test, which basically means that we injected a glucose load and we've taken blood samples for the next 4 hours after that. A total of 26 blood samples in those 4 hours. At the end of those 4 hours, we have taken another muscle biopsy to look at the response or the change in the muscle. So we set some aims for this study. We wanted to investigate the effect of these two different size protein meals on glucose control. Remember that we had the third one as a control. And we wanted to examine if the high protein meals, they alter the activity of protein of interest, so what I was talking there about AKT, relative to a moderate protein meal. And we hope to provide a more comprehensive understanding of the mechanism surrounding amino acids and insulin resistance. And by doing all of this, we're hoping to lead to a more targeted and efficient treatment for those that are at risk of progressing from prediabetes to overt type 2 diabetes. So now let's go and check out some of the results from my study. We're initially going to start with a skeletal muscle analysis. So the muscle biopsies have been analyzed, and we're going to start off by looking at IP6K1. I haven't mentioned IP6K1 so far, but you should know it's a protein that is involved in in insulin resistance development. Our study showed, so it is correlated with insulin resistance, so it's one of the bad guys. So our study showed that IPCSK1 in the muscle was reduced after the high protein meal dose, as we can see. So the HPD means the high protein dose. But then we wanted to see if this is also related to AKT. And you may ask, but why should it be related to AKT? And to answer that, let's go back to the insulin signaling. So we know that AKT comes from PIP2, but what we also know is that also IPCSK1 comes from PIP2 in some respects. So they overlap in this PIP2. So therefore, a reduction in IPCSK1 may lead to more PIP2 being available. Therefore, it can impact the activation of phosphorylation and then activation of AKT. So we tested how different size protein meals affected AKT as well. And while we did not see any changes in total AKT, when we look at the phosphorylation side, we did see an increase in the ratio between AKT threonine 308 and total AKT after the high protein dose trial. And this suggests an increase in the activation and possibly in the glucose uptake. Moving on to other proteins, considering the time of today's presentation, I won't have time to go through all of my data, so I've just selected a few proteins that I'm going to talk to you about. Another one that we investigated is S6K1, and you may remember that I mentioned it when we were talking about mTOR, because it's showing how mTOR is very much correlated with mTOR activation. We did not see any changes in S6K1 after either of the meals, which suggests that probably mTOR wasn't overstimulated, even after the high protein dose. We've also seen a decrease in AMPK after the high protein dose, which suggests that potentially the glucose load combined with the high protein dose led to this reduction, as we do not see any changes in the medium protein dose or in the other dose when we are comparing it to the baseline. Now I'm going to move on to discuss a bit more about the results from the plasma. So we said we've also taken a lot of blood samples and we've measured a lot of different things. I'm going to start off by discussing the HOMA insulin resistance that we've measured. So this is based on fasting levels. So what we see from our results is that we see the appearance of insulin resistance in our obese individuals that were participants. But while we see this, and of course we don't see any changes between the groups, and we were expecting not to see because we controlled it that way. But while we see an increase in while we see insulin resistance, we do not see a decrease in beta cell function. And this is what we were expecting in a way, because what's normally happening as part of diabetes progression, you initially have the insulin resistance, which then later on leads to beta cell dysfunction. So while we have insulin resistance, the beta cell function is still intact in this population. We then moved on to look at glucose effectiveness as well as insulin sensitivity, and we did not see any changes with either of the doses of protein. So despite the fact that we've seen some changes, some positive changes in IPCSK1, AKT, when it comes to insulin sensitivity measurement, we did not see a significant difference. As well as we did not see a significant difference in the area under the curve for insulin, as well as the area under the curve for glucose. We have also measured branched chain amino acids. We were talking earlier about branched chain amino acids. We've also measured them in our blood for the participants for the whole duration. I'm going to start off by discussing the valine one. So in the high protein dose, the one that you see there with C, In the high protein dose trial, we've seen a significant increase from baseline that is lasting from 60 minutes all the way to 240 minutes, so all the way until the end. That increase from baseline was noted in the high protein dose trial, which is different when we're looking at the medium protein dose, because that increase from baseline only lasted until 120 minutes. When we're looking at a medium protein dose with added fat, we actually don't see a significant difference from baseline. So that means that potentially the added fat has an implication in terms of the branched chain amino acids plasma level. And why is that important? Why, or what does it mean to have a more prolonged plasma level of this valine branched chain amino acid? Well, part of this catabolism, so part of breaking down these amino acids, we are producing different intermediates. One of them is, for example, 3-HIB. And this is associated with atherosclerosis and development of type 2 diabetes and other metabolic conditions. So potentially having this prolonged time of increased valine is not a positive thing. But I should mention again, we did not see a significant changes in insulin sensitivity, but it could potentially lead to something like that. Let's see what's happening in terms of leucine. Leucine was the amino acid that was the most potent in terms of activating mTOR. With leucine, again, we see significant changes in the high protein dose when comparing to the baseline, starting from the 60th minute after the meal, all the way to 180 minutes. Which again is different when we're looking at the other two trials. We've seen increases in the medium protein dose, only between 60 and 80 minutes. And for the medium protein dose and added fat, we did not see significant changes from baseline. So again, this is showing that the high protein dose leads to a more prolonged increase in the plasma branched chain amino acids of this specific one. We've also measured the total branched chain amino acid concentrations, and again, we see similar changes. So the conclusions for the first study, the more acute study, is that the high protein dose led to a decrease in muscle IP6K1, which could be a positive. It also led to an increase in the ratio between AKT phosphorylated at threonine 308 and total AKT2. But it had no changes in insulin sensitivity. And we also see some potential negatives because the high protein dose led to a more substantial and prolonged increase in some of the branched chain amino acid concentrations, as we've seen. Now I'm going to move on to discussing my second study, which is following the same, in a way we're looking at very similar proteins, but in this one we have some additives. So first of all, it's a more longitudinal study. So the title is the effects of one-legged resistance exercise training with and without protein supplementation on the insulin-stimulated glucose uptake pathway in skeletal muscle. So here we also have the exercise component. So for this study that was done in collaboration with University of Exeter, participants were divided into two different groups. They either had the protein supplementation or they had placebo supplementation. They didn't know which one they were having, so it was blinded. And they had this supplementation twice a day for 10 weeks. And during these 10 weeks, they've also undertaken 30 sessions of unilateral resistance exercise training. And the key part here is the unilateral part. So they exercise only one leg, but if you look below, we have taken muscle biopsies, or they have taken muscle biopsies from both legs. So we could see what's the difference between the exercise and the non-exercise leg. So a pretty interesting model to work with. We also have blood sampling before and after to see changes in terms of glucose control and also some amino acid concentrations. There's been dietary analysis throughout the study to ensure that the protein that they're intaking is not changing significantly throughout the study. So the aims for this study was again to investigate the effect of protein supplementation on insulin sensitivity in a longitudinal manner, so over a period of 10 weeks. We also wanted to see if the protein supplementation affects insulin signaling and the proteins that are involved in pathogenesis of type 2 diabetes, such as IB6K1, as we were discussing earlier. And then we wanted to see if exercise could potentially mitigate some of the negative effects of associated with an overstimulation of the mTOR-6K1 pathway, if there was one. So again, we're going to move on to see some results from the muscle. So as we've seen, it's a pretty complex model to work with. So I'm just going to initially discuss about what the graph actually means before we move on to the actual proteins. So you will see the black bar is the baseline. And you will see with orange, we have the protein, the group that was ingesting protein. And with blue, we have the placebo group. So we've done several comparisons. We looked at the change from baseline. We look at the difference between legs. We look at the difference between groups. So I'm going to start off with IB6K1. And we see that the cumulative effect of both exercise and protein supplementation is the only way in which over the 10 weeks we see a significant decrease in muscle IB6K1. So with just exercise, despite the fact that we visually see a decrease, is not a significant decrease. But when we add the protein, we see that significant decrease. If you look at the figure, we also see differences between legs in both of the groups. So in both the placebo and the protein group, we see differences between the non-exercise and the exercise leg. And in the non-exercise leg, I should have also mentioned that the U stands for untrained leg and the T is trained leg. So there's an increase in the untrained leg compared to the trained leg in IB6K1. And this suggests that potentially exercise training may have a different effect on the untrained leg compared to the trained leg. So let's remember that increase there as well. We then move on to other proteins, and we've also looked at AKT. And in terms of its phosphorylation, we've seen a difference in phosphorylated AKT at serine 473. Only in the untrained leg of the protein group, we see a decrease from baseline. So that could be taken as a negative. So the group, or the leg, I should say, that did not exercise but had the protein supplementation, saw a decrease in this ratio between phosphorylation of AKT and total AKT. This may mean that it could eventually lead to a decrease in glucose uptake in the skeletal, because AKT is involved in glucose uptake in the skeletal muscle. In the sake of time, again, I'm not going to discuss all of the proteins, but we have analyzed a series of other proteins as well. I'm going to move on to the plasma results. And here again, we've measured glucose, we've measured insulin. What we noticed is we've seen an increase in the HOMA insulin resistance in the placebo group, which was also supported by an increase in fasting insulin. So the change was also from baseline. I don't have the figure showed here, but the change was also from baseline. So that means that the group that was exercising but was not having the protein supplementation, they experienced an increase in insulin resistance as well as an increase in fasting insulin levels. And we have some theories to explain that. So normally when we are exercising, we are increasing the muscle protein synthesis pathway as well as the muscle breakdown pathway, or not the pathway, but muscle breakdown rates. When we are eating protein, we are basically mitigating some of those negative effects of exercise. But when we don't have protein, it becomes a bit trickier. From data published as well, elsewhere by the same group, so from the same study, we know that there's been a difference in the muscle protein synthesis rates between the protein group and the placebo group. So the protein group had a significant increase by 33% in this muscle protein synthesis rate. But both groups led to a 5% increase in muscle volume. So with the differences in the protein synthesis rates, but they led to a same increase in muscle volume. So let's remember that, because it could explain the fact that we see an increase in fasting insulin, because insulin, apart from its well-known effects on glucose, it also has the ability to help with this. It has also an anabolic effect, so it helps with the protein synthesis. So perhaps this increase in insulin or fasting insulin is just a compensatory mechanism to still support that muscle protein synthesis. And there are some studies supporting this. So this is our hypothesis in terms of why we see this change in the placebo group, but we don't see it in the protein group. We have also analyzed plasma IP6K1, and we did not see any significant changes, despite the fact that we've seen some changes in the muscle, but the plasma one was not changed. Very briefly, because I know I'm going a bit too long now, very briefly on the branched-chain amino acids, as we've measured those as well in this study. We do not see significant changes from baseline in any of the branched-chain amino acids or their total value or the combined value. However, when we calculated the delta, which is basically looking at the difference between post-intervention and baseline, we see difference in individual catabolism of those branched-chain amino acids. Because in the protein group, as we see with orange, we have a tendency for it to be increased. Whereas in the placebo group, we have a tendency for it to be decreased. So therefore, this difference becomes significant. Well, that is again just showing us that the protein support promoted a better protein catabolism, leading to those changes in skeletal muscle, volume, and everything else that was better supported by the protein ingestion, but not in the placebo group. So conclusions from this study is that the combination of exercise and protein supplementation led to a decrease in muscle IP6K1, so potential positive. However, there were some negative things as well that we understood better from this study, is that the exercise without the protein support may have led to some detrimental effects when it comes to glucose control, as we've seen in the HOMA insulin-resistant measurements, and also the fasting insulin changes. And also on the other side, if we have protein supplementation, but without the exercise, it can also lead to detrimental effects, as we've seen from this study, mostly related to the AKT activation or decrease that we've seen in the ratio between phosphorylated AKT and total AKT. So we just need to be cautious about what we're thinking in terms of protein and exercise and how we combine this to people. And with that, I would like to thank you and invite you for questions.
Anyone got any questions from the audience?
Yeah, hi there. Yeah, so I'm quite unfamiliar with the mTOR pathway. So I just wanted to check: is the suggestion that when there's high plasma levels of amino acids, that the mTOR is basically stopping glucose entering the cell?
Yes, it's rather more complex than that. Yes, if you have a prolonged branched-chain amino acids in the plasma or an continuous stimulation of that mTOR, it may lead to IRS degradation over a longer period of time. So it's not something that we expect to happen on one day, but we're just talking, like we're talking with hyperglycemia, over prolonged periods of time.
Right. So it's interesting because I kind of, it sort of feels like I was just wondering if there was a sort of like an evolutionary sort of hypothesis of why that's evolved, basically, because it sort of seemed to me that when you'd have a higher level of plasma amino acids, what you'd also want is for the cell to relatively start bringing sort of an energy source in, so that it can start really laying down, you know, muscle fiber proteins and things like that. Is there any thinking as to why it's sort of set up in an opposite way?
It's more likely a combination of different factors, and probably mTOR overstimulation is one of them. But probably there's also a deficiency or inability of the cell to do what you just mentioned, so to use up all of those amino acids.
Right.
Like it's happening in the glucose sense as well. The cells become insulin resistant, therefore they are unable to utilize that energy that is there, but they can't use it. So I think, similarly speaking, this is how it's happening with mTOR as well, from different perspectives as well. So that's just one side of the picture.
Right. I see.
I hope I answered your question.
Yeah.
Yeah, I think that's a good point. You've got me thinking now. I think one thing we need to be aware of is we're looking at, or Oana was looking at, skeletal muscle signaling at a snap time point, and then relating that to whole body measures of glucose control.
Right.
And you know, there's a bit of a limitation there.
Yeah.
But also, what we don't know is what the relative energy content of the cell was. So we don't know whether there was sufficient glycogen or you know, other processes that could compensate from that. But what we were trying to do is unpick whether, you know, if you've got an overstimulation of some signaling pathways on a traditional mTOR or IGF signaling kind of perspective, does that interfere with anything on the other side?
Right.
That was loosely. That was what, you know, we were into a PhD. Obviously, Oana has what she wants to do, and then she has supervisors that want other things from the PhD.
Yeah.
So. Yeah. Yeah, but it's a really good question. Something Alex's PhD student is still trying to unpick as well.
Thank you for the question. Any other questions?
Anyone from outside? You could ask them. You've got a little bit of time. No? I've got a question. Maybe you can go back to your slides.
Go back to the slides.
Not a difficult one. It's more probably I was running around. So as you go down to the conclusions, trying to stop people from coming into the room.
Conclusions of.
Yeah, this one. Yeah, it's probably more around. You know, perhaps just trying to unpick this a little bit more, because I you know, I've read the PhD, but I'm still not quite sure, you know, how that might be working at a whole body level in terms of this, you know, the last conclusion.
Which one of
This,
them?
the last one. Or both, you know, if you want to answer them both at the same time. I'm just not sure I really followed exactly what the conclusions, what, you know, the reasoning behind the conclusion, if that makes sense at all.
So there are two different ways of, two different conclusions in a way. So for the protein supplementation without exercise, we were just looking at, and I can just go back to, we were just looking at the AKT phosphorylation. And then when we analyze the untrailing, but with the protein supplementation, we see a decrease in one of the phosphorylation sites compared to the total AKT. So therefore, that could potentially lead to a decrease in glucose uptake. So those are the type of detrimental effects that it could have on insulin signaling when we have maybe an excessive amount of protein ingested and without the exercise component, because we do not see any changes in the phosphorylated AKT over total AKT in the train leg with the protein supplementation. So that means there's something else going on when we potentially have maybe too many amino acids.
So, in short, if there are lots of amino acids, you make sure you exercise. Otherwise, if you're eating a lot of it, you might have negative consequences.
Exactly.
Okay, I did follow that. Thank you. Thank you.
I've got a couple of questions, Oana. Just first of all, if you wouldn't mind, just remind, you probably said it at the start, and I probably was not quite here yet. But just how many participants you have in both of your studies?
So for the first study, we had a total of 9 participants, and for the other one, we had a total of 16 participants, which was again divided into 2 groups.
Okay, great. And they were all healthy?
In the first group, no, we had differences. So in the first study, we investigated these effects in obese individuals, So that means a BMI over 30 or body mass index over 30. And in the second study with a unilateral exercise, it was done on healthy individuals. So yes, it's a good point because we do need to take into account the difference in their metabolic status to begin with. So while we can compare these results to some extent, we need to consider those metabolic changes. So one of the things that I would like to do further is to be able to replicate some of these studies and investigate it into different populations. As I've mentioned at the beginning, metabolic state as well as physical activity levels and a lot of other things need to be considered when we are discussing about this.
Yeah, great. Yeah. So my next question is, so you ended up concluding with the first study that even though you saw changes in phosphorylation of AKT, it did not translate into changes in insulin sensitivity, as you very nicely just and very clearly put up there. Why do you think we don't see that translation?
That's a very good question. Thank you. I think, first of all, it was an acute study. While maybe we start to see these changes in phosphorylated AKT, again, we did not see any changes in total AKT, and it is known that it takes a longer period of time for changes in total protein to appear. So probably time is one of them. Why we did not see. Another one, it could be that we just did not reach a threshold in order to make those specific changes. We did not see any change in N6K1. So perhaps even with the high protein dose, we did not overstimulate mTOR as we thought we might do. Yeah, I think these are two reasons, and I can probably think of more.
I think there's one more, which is the analysis still to be, so we're mathematically modeling the labeled components, so to look at disappearance rates in the whole body. So when you add that other variable in, you might see differences, but I'm probably leaning towards what Oana said is not long enough and not big enough dose to really see any effect in the short term.
And then I have the last one. This is very specific, I guess. On your graphs looking at your amino acids, for example, valine was it? Or,
yeah.
Where you saw that with the high protein branch of your protein, or a prolonged increase, whereas you didn't see it with the two other groups.
Correct.
But if I may just be the devil's advocate here, isn't that just because you have a high protein intake? And the question then is, if it's not just because you have a high protein intake, for other amino acids, do you not see that increase?
I haven't specifically measured all the other amino acids. I've focused on the branched-chain amino acids for these studies. And yes, in a way, I see your point. With a higher ingestion of protein, we are expecting in a way an increase. But we wanted to confirm that, and we want to see what's the implication of that. What does it mean? And I think that's the trickiest part, and that's when it comes, okay, if we're eating this much, what's the consequence of that, and how we can translate that into the insulin signaling, and how we can translate that into diabetes progression. So that's the part that we try to focus on. Yeah. Sometimes it's not, perhaps just don't eat that much is not enough. We need to understand why we shouldn't be eating that much, and how is it affecting us, or how is it affecting the people that are in this process of transition from prediabetes to diabetes, perhaps.
What do you think? So you check the branched amino acids. Which would be the most dominant amino acid to maybe just check as a marker, for example, do you think?
Sorry, which one?
Would there be a sort of common amino acid in the diet that you could check as a marker? If you're going to, continue to investigate this a bit more, what could you possibly do as a kind of standard to see whether this is just what you see, generally speaking, or because if it's not, if this is particular for some amino acids that we maybe know are particularly detrimental, then that would be interesting and important to know.
I think it's not only limited to the branched-chain amino acids. I particularly chose the branched-chain amino acids because they have been linked, because their increased level of fasting levels have been linked with insulin resistance more particularly. But these are not the only ones. I would measure all the other amino acids, and probably we would see more changes in there as well. And there was, I think it was tyrosine that had influence on other things as well, such as the hormonal response in terms of how we digest, so all of those digestive related hormones as well. But it's a bit out of my topic, but I'm gonna read more about it because it's a key point, I think.
That's a good question. It's not something we've thought about as a potential, whether there's a one or two amino acids that could be particularly early signs of, issues. I guess that might be Deanna's work, which has the longer, the 9-month diabetes program, that we might see some of that data. I think with Oana, we felt at some point that we couldn't make that jump from just eating and measuring. It's about measuring what was actually amino acids. So it's more of a, well, we had to, we wanted to measure it to make sure that what we were doing was actually increasing amino acid availability to then make the next step. So we haven't really thought about that at all. But it's not bad. And did I hear you say you're going to do more work? That was the opposite of what I was thinking. You said when I can. So I've been waiting for about 10 minutes to finish asking your questions before I brought that up. I look forward to hearing that. That'd be really, really good.
See you on Monday.
Yeah.
In the lab.
Yeah, I mean, I think if Nick Curran was here, he would, if he had his way, we probably would have measured every amino acid and some others that aren't available currently. But thankfully not. I don't have any more questions. Anyone? Yeah.
So one last question. First of all, congratulations on getting to the end of your PhD. That's obviously a lot of work. I mean, I've seen you run around with your clipboards, but I think now I can finally see just how much work has gone into it.
Thanks so much.
My question is very basic. I know nothing about insulin resistance, so it's just a methodology question. With your study that was going across the 12 weeks, it was a unilateral training program. I was wondering what that was like in terms of the participants. So did they just report that they did only training for that one leg, or were they training any other body parts at the same time?
The training was completed in the lab, so it was under supervision, and they only trained that particular leg.
And they did no other training just like outside of the lab sessions?
No, it was monitored training.
Okay.
So it was another part of the question?
No, it was just that I was just wondering in terms of like a
yes, it was monitored, so it was a training that was monitored, and then they had the protein at home. I mean, they had protein after the training on training day, but of course they didn't exercise every day. And if they didn't come to the lab to exercise, then they had protein in the morning at home, and then again in the evening. Another set of as a supplement, yes. But it was again double blinded, so they didn't know whether they were having the protein or the placebo supplementation.
That side, there's another side to the EXTRA study that is published. If you wanted to read more about it. So
I still have no idea what AKT or any of the other letters were. So
do you think I do?
No one does. It's like the rules to rugby. No one knows what's going on at all. I think Oana's review paper is a good starting point to understand the mechanisms. But really, the other study was looking at the direct effect of the supplementation on protein synthesis. So that's why they're a very complex study design with lots of muscle biopsies.
Yeah.
Lots more.
It just fit quite well with our research question, and we decided to do some further analysis. Thank you for the question.
Thank you. Any more quick questions? No? So, yeah, thank you again, Oana. Can I lock the door now? I may be so angry. I almost tripped over the stairs running down the second time. That would have been really good.
That's the reverse seats, isn't it? That students felt they had to come in to tap their cards because the rooms had changed.
I know.